Hey, I'm JT Stone.
I'm Daishan Line, and we're here at Cornell University.
Today we're here to meet Laura Schoenlein.
My name is Laura Schoenlein. I'm an Interfaces of Global Change Fellow at Virginia Tech. I'm also an EPA STAR Fellow. But I am doing research on things that I'm interested in, on things that in particular actually the EPA has funded me to do. What I study is how the hormones that are associated with stress affect the way animals deal with disease. And that's important because you could imagine that as the world changes, things with like pollution and even like noise pollution, like car traffic and people expanding their, the human habitat expands, animals might get stressed out by that. And so when animals experience stress, just like when you experience stress, these hormone levels go up. So I'm trying to understand how these stress hormones affect birds that have avian malaria. I work, actually most of my field work is up in Canada, in southern Ontario. I go out and I catch birds, I take blood samples. I have to do it really fast because it's stress hormones, so if you wait too long, their stress hormones go up because they're stressed out. So you have to catch them and take the blood sample in less than 3 minutes. But I spend a lot of time just kind of walking around outside, carrying stuff. I walk through marshes, I have chest waders. Right now, I sit at my computer all the time. I'm writing a paper and I'm doing computer coding.
Do you enjoy your job?
I love my job.
If you could change anything about your job, would you change anything?
Yes.
What?
I would change stability. The job market is kind of unstable, and my funding actually comes from the government. And the current president is not a big fan of science funding.
What kind of equipment and materials do you use to test the animals?
Yeah, so I catch them in these nets. So I put this dead bird on a stick in front of the net, and I do it on another bird's territory, and they get so mad, they fly at the dead bird, and when they try to fly and attack him, ideally they hit my net, and then I can run and get them out. I use like needles to collect blood samples. I use speakers to call the birds in, so I play back their calls to them, and they'll come in. Oh, binoculars are probably my most important tool. And, this is stuff that we use to do the lab work, to figure out how much hormone is in the blood. So this is a centrifuge. You put the blood in a tube like this one, and then you put the tubes in here, and when you centrifuge it, what it does is just spins this thing really fast, right? Mm-hmm. And so the heavy part of the blood, which is the red blood cells, sinks to the bottom. And the light part, which is yellow and kind of clearish, is where the proteins are, the plasma, that stays on top, and that's where the hormones are.
Those are micropipettes?
Yeah, do you guys have those?
I just used them.
Oh, cool. Did you do a gel? What are you doing?
Yeah, gel. I forgot what it was for, but yeah, we were testing blood samples.
Yeah, the gels show you DNA. For me, I tested if the birds have malaria or not using gels like that. I do it's called a PCR reaction. It's basically you make lots of copies of DNA and then you put it in the gel. And if they have the parasite, the reaction makes a lot of copies. And if they don't have the parasite, there's no DNA to copy, and so it doesn't show up. So when you put it on the gel and you can see the things light up on the gel, you know that parasites or not.
So stuff we do in school actually happens at Cornell?
Yeah, the actual methods that you did are exactly what we do. So this is all hormone stuff, assay stuff. Basically, this thing is a vortex. You can feel it. See if you want to see what it feels like. You can just do it, and it mixes it up for you.
Oh, that's pretty cool.
Yeah, and so you can imagine if you have liquids in there, it makes almost like a tornado inside there. It just swirls everything up. My favorite and least favorite piece of equipment. This is my microscope. Yeah. So I look at the blood of the birds on the glass slide and I count how many infected cells there are.
What would you say your greatest strength here is?
I've been working a lot on science communication and improving my writing and speaking. I think I'm getting there. I don't know if it's my greatest strength, but I think it's an important strength because obviously people don't really understand. You hear all this climate change debate stuff happening, and everyone who's a scientist knows that there's no debate. Climate change is happening, it's caused by humans, we have all the data, it's true. But clearly as scientists, we're not doing a very good job talking to people and getting them to understand, what we mean. And it's so important that people understand. And people who aren't scientists are equally as smart, they just are smart about other stuff. And so we have to find a better way to communicate with them.
This is one of the first times I've actually been to a Cornell location before meeting an actual student. And I guess it's just really interesting to, learn what they do there, and especially in the field of science, and learning that what we're doing in school right now does apply to the real world.
Things that we learn in school in biology actually do apply to real life situations, and it's important for us to know that because biology is changing the world one step at a time. Don't forget to subscribe.